Lonneke. Je staat op mute.
Ja, ik heb hem gevonden.
Ga je nog, moet er nog iets van een voorstelrondje gedaan worden of zo? Of ga je gewoon degene aankondigen die gaat praten?
Ja, ik stel iedereen voor.
Oké, top. Zal ik dan weer een mute en de camera uitzetten?
As many people around, so hopefully have. Yes. Oh, we should mute our mic.
Please turn off your microphones. Thank you.
I will wait another 2 minutes because there are only now 25 people online.
It's a great idea. Thank you.
Boop. Boop, 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 boop. Putain, mais merde.
Lonneke, maybe we could start. We are now 40 people, and I think people will be joining us relatively soon.
Yes, so everybody welcome to this webinar on understanding Opsin-Related Retinal Disorders. For the next 1.5 hours, we were talking about one of my favorite subjects, the Opsin gene cluster. This webinar is divided into 4 parts. So first of all, Dr. Renata Sarno will present the patient perspective. Then Dr. Susanne Yzer will present the clinical perspective. Thirdly, Dr. Susanne Kohl will present the Opsin cluster and standard diagnostic approaches. And then finally, I myself will present novel diagnostic approaches. So this first session, where the patient perspective will be covered, is presented by Dr. Renata Sarno. Dr. Renata Sarno holds a Ph.D. in theoretical physics and is based in Italy. She has 5 relatives affected by blue cone monochromacy and has been a dedicated patient advocate for BCM and achromatopsia since 2009. And Renata founded an international community of individuals and families living with blue cone monochromacy and serves as a president of the BCM Families Foundation. So Renata, you can share your presentation and start.
Thank you very much for the introduction. I'm trying to share my screen. Okay. And thank you for the opportunity to speak about blue cone monochromacy. I'm here today because in my family there are 5 members affected by blue cone monochromacy. And also because I'm the president of the BCM Families Foundation, an international community that collects more than 200 families affected by BCM internationally, and around 800 patients affected. And what is blue cone monochromacy? Is a rare genetic eye disease affecting 1 individual in every 100,000. And is a debilitating disease that is a lifelong disease. What is exactly BCM? In the retina, there are eye photoreceptors, and patients affected by BCM have red and green cones, but they are not functional due to the lack of critical proteins, the opsin proteins, the one we are speaking about today. This means that the central vision of people affected by BCM, the daylight central vision, is due only to blue cones that are only the 5% of the cones in the retina. And today I would like to tell you the story of the patients in my community and the story of my family. In my family, blue cone monochromacy started in 1899 when my grandfather was born, and he was affected by this disease, but he didn't know the name of the disease for the entire his life. Then other people appeared, other men appeared in the family with the same disease. And nobody knew the name of the disease until when I tried to find a diagnosis for my son. And in 2009, at the end, I reached a clinical diagnosis and a genetic confirmation thanks to the Tübingen lab. And so there were 110 years before between the first people who appeared in my family. And what is the diagnosis for a person with blue cone monochromacy? What we know from our community is that the diagnosis is an odyssey for a patient with BCM. And we estimate that 1,000 of patients are without a diagnosis. Many of them arrived after a long story of misdiagnosis or with a long period of undiagnosed, in which the disease was undiagnosed, and with a very long time in which they tried to obtain both the clinical diagnosis and the genetic confirmation. And what is the diagnosis for a patient? For a patient with a rare disease, the diagnosis is a relief because you finally give a name to the disease that is running in your family. And is also a milestone in the life of an individual with BCM, because you can start planning and doing other activities, participating with your community, supporting the cure, or just planning your life. So my advice is to give to patients their own DNA test report, because this is a part of their identity. And the story inside other families of our community is exactly the same. As you see, there is a grandfather affected, the female carriers, and then a cousin or a brother, and the son and grandson. So we collected at this point 79 families inside our international patient registry. And we have the family trees of all those people. And we know that there are large families also with more than 20 people affected, some others in which the disease appears after several generations. And the average number of people affected in each family is about 4. And this is the reason why we call it the foundation BCM Families Foundation. What is the first symptom that a person with BCM show? It is nystagmus, and we see nystagmus in the eyes of our children when they are 6 to 8 weeks old. This is newborn children. So we see nystagmus, the appearance of nystagmus, and this is the first symptom that a female carrier see in the eyes of his baby. These symptoms remain also in adult life. This is an adult with BCM. And we estimate that around 1/2 of the people in our community has nystagmus in adulthood. And this is a lifelong disease that starts from birth. And for the entire life, individuals with BCM experiences low vision, nystagmus, poor color discrimination, myopia, and extreme light sensitivity. I want to tell you that these people have never experienced the normal central vision, so they don't know what it is. Having followed a child with BCM, I know that he discovered step by step what does it mean not having central vision. So I'm giving voice today to their silence, because they don't express, okay, I lose something, because they don't know what they have lost. And the most important symptom for people with BCM is low vision. Here you see a table coming from a clinical study that our community of patients financed and then participated to. It was a large collaboration, and Samuel Jacobson was the principal investigator. And here you see that there is a collection of visual acuities of people with BCM. There are people that are legally blind, and there are also people with a milder, less severe form. And in any case, this is the most important symptom. And we see that our children and then adults bring toys and books and objects very close to their face. The other aspect is the poor color discrimination. They mix black and red, yellow and green, pink and blue. We did several tests between us, and we have demo glasses that has been prepared by a member of our community, Dean Montey, and to show what is the color vision of people with BCM. It's a mix of blue and green, I think, something like that. The other point is the photophobia. This is also debilitating, and people without a filter, without sunglasses, have usually this squinting expression. And also there are people with BCM have a collection of filters that bring with them that are brown, magenta, and red. One of the biggest challenge of BCM is social and not visual. This is one of our testimonials telling that people tend to see different behavior as reflecting intelligence or personality rather than as a result of physical differences. And the main reported social problem in our community is not recognized, for example, the faces of people around you. And this is a cultural problem because low vision is unknown by the general public. So they tend to don't think that you have a physical difference. In the life of people with BCM, there are many obstacles, and one is orientation and mobility. For children, they get lost in unfamiliar places or outdoor, hit obstacles, and need orientation and mobility programs. In absence of a diagnosis, is very difficult to make all the decisions that are important for the life of these people. Also, living in a place where public transportation is not accessible could be a problem because you don't see, for example, signs or you have bad accessibility of public transportation. Finally, also the driving license, because people with BCM, achromatopsia, or ocular albinism, and many other central vision problems, diseases, can drive in 48 to 50 states in the US. But in Europe, there is only one country, the Netherlands, that allow people since 2008 to drive with the bioptic telescope. And we did a lot of efforts with the Achromatopsia Association here in Italy to export this beautiful the Netherlands program, but we couldn't make it so far. Having a diagnosis is very important if you have children with BCM, because they work with the teacher and yes, with teachers, parents, and children at school is very hard. And to explain exactly to teachers what a child has is very, very important. You need to review all materials, book, picture, maps, whatever, find digital books, enlarging text. I had a photocopier machine at home to enlarge everything. You need to managing the fatigue because children with BCM are very slow in reading, and those who have nystagmus in emotional situation have a lot of problems more. So we developed, for example, best practices for school integration, but this is something that we share with people that are in contact with us, that knows the diagnosis. Also for the selection of the job, it could be better to have an earlier diagnosis, and we have a program inside the community where people can share their own experience. Older people can tell to younger what was their life and their experiences with job. And so this is a way to share experiences and to make a selection, a better selection. Also, sport could be a challenging, and also here the diagnosis could be important. So this is the story of a single patient or a single family. And then since 2009, I decided to start a community of patients because I felt so alone without knowing no anyone else with the same disease outside my family. So we started with other families, the BCM Families Foundation, and this community is today something that answers to some problems because BCM is a rare disease and families are few and far between. They feel a sense of isolation. They have day-by-day issue, especially for with children. So we give a community with meeting, newsletter, social event and networks, and we have a program for DNA test, a program to refer people to expert ophthalmologists. And our way to fight BCM is to concentrate information about BCM in some expert centers and in our BCM patient registry. What this community wants? We want a cure. This is our mission. We want a better diagnosis, and we want to improve the life, especially in Europe. This is a picture that is coming from one of the clinical studies that has been done on blue cone monochromacy. We supported this study both financially and also participating to this study. And you see here a summary of what is going on with deletion and C203R deletion, that are the main causative mutation for blue cone monochromacy. You see that in the first, second, third, and fourth decades, there are possibility to make gene therapy to those people because photoreceptors are still there. Then there is a degeneration. So we participated to clinical trial. I would like to show this one. We collected and sent more than $3 million to finance natural history studies and to prepare outcome measures for a clinical trial and to test several AAV vectors. And also we worked with the Tübingen lab in order to have all people inside our community fully tested, genetically tested. This is our scientific advisory board and the board of directors. And I told you that we want a cure because last January, I prepared a letter for receiving a grant, and I asked to my community to sign the letter. And in few days, I received more than 400 signatures, that many of them with sentences that expressed the urgency and the need for a cure. And obviously, if a cure there will be in the future, that is our hope, and we think that the diagnosis will be very important at that point. I want to tell to all clinicians and patients that we have a patient registry, and they are all invited to support this project with us because it's our way to fight blue cone monochromacy. Concentrating all information about this disease is we feel that this is our way to fight BCM. These are our testimonials. Some of them are very young, and I want to thank you for this opportunity.
Thank you, Renata, for your compelling presentation. Are there any questions? I see a question in the chat, but I don't think it's for Renata herself. Maybe for Susanne Yzer in the next presentation. Are there questions for Renata? If not, then we go to the second session. Oh, it is for Renata. Okay, so the question is, on the OCT images, some of the scans seem to have no foveal depression. Do you feel that there is a degree of foveal hypoplasia?
I don't think that I'm able to answer to this question. Maybe a clinician can answer better than me.
Okay, I will keep this question for the next after Susanne Yzer has had her talk. So the second session, where the clinical perspective will be covered, is presented by Dr. Susanne Yzer. Dr. Susanne Yzer is a board-certified ophthalmologist and a clinical scientist with expertise in retinal care, ophthalmic genetics and translational therapeutics. At Radboudumc, she leads the medical retina and ophthalmogenetics team, overseeing gene therapy trials and mentoring Ph.D. candidates. The floor is yours, Suzanne.
Thank you, Lonneke. I hope you can all see my screen now. Is it working properly?
You're not sharing your screen yet.
I am, but apparently something is not going right.
Just now I can see your files.
Now?
Yes.
Okay.
On your screen.
So thank you very much, Lonneke. So I was actually specifically requested to talk on Bornholm eye disease, and I was very, very happy with all the nice information that was just shared with us on BCM. I have no disclosures. So Bornholm eye disease, it was first described in a Danish family from the island of Bornholm in 1988. It's a rare disease, but honestly, I think their prevalence is unknown and it's probably underestimated. And I hope that through this talk, I'm able to explain to you why I think that's the case. So in this presentation, I will cover the X-linked inheritance, the cone dysfunction, dichromacy, myopia, and imaging on Bornholm eye disease. So how about the X-linked inheritance modus? So as you know, males have an XY, so they only carry one X chromosome, and females are XX, so they have two X chromosomes. And that means that if a carrier woman, she has one, let's say, benign and one pathogenic allele. So she's only, she's not affected or only mildly affected, and she will pass either the pathogenic allele or the benign allele. So affected males, they have inherited this pathogenic allele, and they don't have a second X allele to make up for that. Affected women also exist. That's very, very rare. They have two pathogenic alleles. So in X-linked inheritance disease, affected males cannot pass the traits to their sons. Affected males will pass the pathogenic allele to all daughters, and daughters will be carriers and are not or only mildly affected. Carrier females have about 50% chance of passing the affected gene to each child. That means that sons with the pathogenic allele will be affected, and daughters with one pathogenic allele will be carriers. So what the main thing is, what will guide you to X-linked inheritance is when there is no male-to-male transmission of the disease, and that's shown in this example of a pedigree. Now, how about cone dysfunction? In the retina, we have opsins that are essential for the visual transduction cascade. And in rods, these opsins, this opsin is rhodopsin, and this only functions in dim light conditions. In cones, we have three types of opsins, and they are important for color vision, but also for detailed vision, so visual acuity. So these three types of opsins that we know are the short opsin, the S-cone opsin, the medium opsin, the M-cone opsin, and the long opsin, the L-cone opsin. The combination of signals from these different cone types allows us to perceive a wide spectrum of colors. And the medium cone opsin and the long opsin, cone opsin are, these proteins are generated from genes that are located on the X chromosome, so what we've just discussed. So pathogenic variants in these two genes are causative of Bornholm eye disease, also of BCM actually. And their underlying mechanism is very complex because they form a cluster, and I'm sure that Lonneke will talk about that in more detail. So mutations here cause a reduction in the levels of normal opsin and abnormal cone function. So this leads to reduction in color vision and reduction in visual acuity. And like I just mentioned, that mutations in the same cluster are also linked to other phenotypes like BCM. We also know from ERG recordings that there is indeed cone dysfunction. So scotopic waveforms are normal in Bornholm eye disease, and these are rod-mediated, rod-dominated. Photopic waveforms in these cases are subnormal or low, and these are cone-dominated. So studies have been performed in carrier females and unaffected males that their ERGs are normal. Here's an example of a normal male control for mixed rod and cone function ERG, and here's an example of a patient with Bornholm eye disease. And you can see that especially this 3 is lower, so it's subnormal. Here the ERG recordings of the maximum flash photopic, so this is for cone function. And again, there is the normal male control versus the affected patient by Bornholm eye disease. And you can see that this entire region is like almost flat. Now, how about visual acuity? We've studied 40 Dutch patients with Bornholm eye disease, and their visual acuity ranged from about 10% to 80%. This is Snellen visual acuity, with a median of 30%. And it's shown in this graph here. And as you can also see, is that most of our patients are relatively young. So we don't really know what happens to this visual acuity later on. But it appears, because each line represents follow-up in a single case, that visual acuity improves a little bit in the early years. But most patients have vision that's lower than, let's say, 55%. Well, how about dichromacy? Dichromacy means that you have two functional cone types. And what was already mentioned in the previous presentation is that the patients with BCM have color vision defects, but in Bornholm eye disease as well. And if we just step away from Bornholm eye disease and BCM, that in the general population, 6% of males have a deuton, that's a red-green color vision defect, which is caused by reduction in the sensitivity of the green light, so that's by the M cones, and about 1% have a proton visual defect, a color vision defect, which is a red-green color blindness, which is caused by reduction in sensitivity of the red light caused by L cone abnormalities. So this is what's in the general population. This is not linked to BCM or Bornholm disease, and these patients don't necessarily have myopia, and their ERGs are normal. So what was found in the patients with Bornholm eye disease? Well, of interest is that both protanopia and deuteranopia were described. And that's not a big surprise if you look at that the M and the L cones are, again, they are in the same cluster. So when we look at our 40 Dutch patients, well, we did color vision tests in 60 eyes, and there was a proton defect in 27 cases, deuton defect in 14, and 12 had some other kind of color vision abnormalities, for instance, by an additional triton color vision deficiency. And of interest is that 7 of our cases had normal color vision. So there's large variation, and that appears to be independent of the genotype. How about myopia in Bornholm eye disease? From studies, we know that myopia is already seen in these cases by the age of, let's say, 1 year and to 5 years. So when they're diagnosed with myopia, they have a dioptic refraction of about -7 to -11 at this young age already. It appears that it's stable after puberty, and what's also described in literature is that their retinal disease appears to be non-degenerative. So it appears to be stable. Adult individuals have an average axial length of about 28 millimeters, which is longer than for an emmetropic eye. So when we look at our 40 Dutch patients, we have refractive error at baseline in 34. And as you saw in the previous image that I showed of our cases, that they're relatively young. At onset, they had a refraction of about -6.5, which during follow-up increased to about -8 in both eyes. Follow-up time was 4 years. And it's shown here again. We mainly have younger patients in our cohort, and the spherical refractive error, so the amount of diopters is decreasing, like as the error just shows. How about imaging in these cases? We saw in our 40 Dutch patients that they have a tessellated fundus, which is also that is compatible with the myopia. So tessellated fundus is, for instance, shown here. So when you can see these large choroidal vessels, which is not the case in an emmetropic eye. They have tilted discs, which is also, as an example shown here, which is also only seen in myopia. They have peripapillary anomalies like atrophy and peripapillary crescent. And there's retinal thinning with aging, and I will show you that in the next slide. It's unclear if myopic complications occur, so it's not been described in literature. Like I just mentioned, we don't have long-term follow-up in our cases, and in literature there are not that many cases been published. So it's of interest to learn if myopic complications occur. So here are examples of the fundus autofluorescence and OCTs in a few cases, and what you can see in the younger cases is that there's central hyper-autofluorescence on the fundus autofluorescence, and in the older case, which is shown at the bottom, you can see that there's central hypo-autofluorescence. And this corresponded to disruptions that we saw in the central area on the OCT, which appeared to be due to the loss of photoreceptors. We also saw posterior staphylomas in our cases, which is a hallmark of high myopia, or pathologic myopia, actually. And here are some other cases that actually represent the same. And you can see at the, if you look at the bottom, that with increasing age, the retinal thins. So we also performed adaptive optics in our patients, at least in some. Here's an example of normal adaptive optics imaging in a healthy participant. And these are representative of the smaller cones. They are very close to the fovea. And in the fovea, you see blurriness due to a high density of cones. So this is in a normal situation. If we look at a case with Bornholm eye disease, you see in the fovea. So this is where in the normal participant, it's all blurry, and this is actually visible in the same area in a Bornholm eye disease patient. So you see all these small white dots, and these probably represent enlarged foveal cones, and they are surrounded by blurriness. And here are two more examples of cases with Bornholm eye disease. So this is like very obvious, but this is not specific for Bornholm eye disease. So similar images have also been made in patients with BCM. So in conclusion, we can say that Bornholm eye disease is an X-linked disease. It's caused by mutations in the opsin genes. The phenotype in Bornholm eye disease is quite variable with cone dysfunction, color vision defects, high myopia, and long-term follow-up data are roughly missing. So it's very interesting to see how these patients develop over time. And what we saw in the previous presentation that this is really different from BCM, although mutations are that cause BCM are in the same genes, but these patients have severe color blindness, they have poor vision and photophobia, which is, especially photophobia, is not a sign of Bornholm eye disease. So that's why I think it's more frequent than we now think, because we see high myopic male patients with suboptimal visual acuity that really function well. So they are not necessarily just. Sorry, that's my Siri. That's why I think it's underdiagnosed. Well, I want to express a special thank you to the patients that participated in these studies, to Timo Mulders and Kyle Hoying, and to Zenita Smilojic, who collaborated on the clinical data and on our cases. Thank you for your attention.
Thank you, Suzanne, for this nice clinical overview. So the question before still stands, so maybe you can answer it. So in the previous presentation on the OCT images, some of the scans seem to have no foveal depression. Suzanne, do you know if there is any degree of foveal hypoplasia in patients with blue cone monochromacy or Bornholm eye disease?
I haven't seen it in Bornholm eye disease. I also in BCM we have not really seen many cases. I've never seen it in any of ours, so that's what I can tell you about that. And the scans that were shown in the previous presentation, I just received a phone call by a patient at that time, so I haven't seen the OCT, so I should have a look at the scans to say anything about that. I'm sorry.
Maybe we can pass on this question. Art Sedation is on the call, I see. Maybe Art, you can comment on whether you ever saw in your BCM patients that they don't have a foveal pit, which is often seen in achromatopsia patients. But I'm also not aware that this is the case for BCMs.
So foveal hypoplasia is variously defined. Usually it refers to the existence of inner retinal cell layers that shouldn't be there at the fovea, and thus flattening out the foveal depression that normally occurs. I do not believe that's a typical appearance of BCM OCT. And of course, there's always exceptions, but not typically. And the other reason one can have no foveal depression is if there is a preferential parafoveal loss, then everything kind of looks flat, because normally the immediately surrounding the fovea is a much thicker retina, and that causes the appearance of a foveal depression. and I don't believe that is typical also for BCM. So the typical, I'm much less familiar with Bornholm eye disease, but at least looking at the OCTs, what appears to be common in both conditions is a much further outer retinal abnormalities that are at the level of the outer segments, which are in these images shown at the bottom, and that seems to be common in both kinds of conditions, BED as well as BCM. So that's kind of a long explanation.
Thanks for stepping in.
Yeah, thank you so much, Arthur. Are there any other questions for Suzanne? If not, we'll go to the other Suzanne.
Yeah, so maybe I can just add that for the OCT, you should really, you can only say anything about it when it's really going through the fovea. So if you just scan parafoveally, it may appear as hypoplasia. So that's why I said I didn't see the images from the previous presentation, so I cannot say anything about that, but that's very important to look at before we can jump to this conclusion. Thank you.
Thank you for the addition, Suzanne. So then we go to the third session where the Opsin cluster and standard diagnostic approaches will be covered. This is presented by Dr. Susanne Kohl. Dr. Susanne Kohl is the Deputy Head of the Molecular Genetics Laboratory at the Institute of Ophthalmic Research, Center for Ophthalmology, University Hospital Tübingen. For over 30 years, she has been investigating the genetic causes of inherited retinal dystrophy. Suzanne has contributed to the identification of numerous genes associated with these blinding disorders, with a particular focus on achromatopsia and related cone photoreceptor diseases. So the floor is yours, Suzanne.
Thank you, Lonneke, for introducing me. I will try to share the right one. So, do you see the presentation?
Yes.
So we will now turn to more closer description of the red-green opsin gene cluster and the genetic causing and of especially blue cone monochromacy, but also Bornholm eye disease and the genetic research testing that we offer in Tübingen. So you might be well aware that we have quite a large cohort of achromatopsia and BCM patients, and to set them into relation in numbers, I want to maybe mention that if we look at the achromatopsia cohort as a whole, including the BCM make about 20% of cases. And we already heard that it is an X-linked condition. Mainly male subjects are affected, but we are well aware of the one or other family where also females present with the cone dystrophy phenotype. And we think this is likely due to skewed X activation, but this has not been studied. As said, it's a rare disease with a prevalence of about 1 in 100,000. That means that in Europe, we should have about 3,500 patients, but the numbers that we have are much, much lower. So blue cone monochromacy, or from now on, I will call it BCM. We already heard there are no red and green cones functional, in contrast to achromatopsia, where none of the 3 cone types are functional. And that is due to the, let me take my pointer, the situation that the OPN1MW and OPN1LW that encode the middle wavelength sensitive green opsin or the long wavelength sensitive red opsins are located on the X chromosome here, down here at the very, very tip. And the third opsin that we have is encoded by the OPN1SW or short wavelength sensitive blue type. But today we're solely focusing here on the red and green opsin gene cluster. These 2 genes, or it's more than 2 genes, are situated on the X chromosome as a so-called head-to-tail tandem array, and they have evolved in recent evolution by gene duplication in the lineage of old world monkeys about 30 to 40 million years ago. And it's important to understand that the OPN1-LW and NW genes are about 96% identical, both on the exonic and intronic nucleotide level. And if we look at the protein, they only differ by about 7 amino acids that more or less give a wavelength shift of 30 nanometers. And that enable us, together with the S-opsin, to trichromatic color vision. If we look at the gene cluster in more detail, we see that this would all be male subjects, have different numbers of gene within the cluster. To be a normal color-sided subject, it would be completely sufficient to have one red opsin gene and one green opsin gene. But actually, many, many more are within the cluster. And on average, there are 3, but within our studies, we have seen as many as 14 genes in this cluster. And thinking of the sequence identity, this brings some problems about, meaning there is non-allelic homologous recombination during meiosis, where the clusters from the 2 chromosomes mispair. If such a mispairing and a recombination takes place intergenically, so between genes, this can result in the loss or gain of gene copies. And if this happens within a gene, so intragenically, this results in hybrid genes. These hybrid genes are associated with the red and green color vision deficiencies. And they are not really like disease relevant because these hybrid genes are perfectly functioning visual pigments. They only don't have those 13 nanometers wavelengths different, meaning that the color vision deficiency is defined by the intermediate absorption spectrum. Looking at the genes within the cluster, we also have to think about the expression of these genes. And it is long known that a single cone photoreceptor, at least in human, only expresses one cone opsin gene, meaning it expresses either red or it expresses green. And the expression of the genes is regulated by an enhancer element called the locus control region, or LCR, which is upstream of the complete gene cluster, and each single gene has an own promoter. In 2011, Peng and co-workers suggested the so-called chromatin loop model for the exclusive activation and expression of red or green opsin. And here we see a depiction how they propose that this occurs. The locus control region stably links to either the L or the M promoter, driving the expression of this opsin in this single cone photoreceptor. It has also been established that the probability of expression drops with increasing distance from the locus control region, and it is well accepted today that essentially only the first 2 genes in the cluster are expressed, red more than green, and that the downstream genes essentially do not contribute to color vision capacities and also typically likely not to disease. Now let's look at the genetic causes of BCM. The first important cause are really large deletions. They can be variable in size. The smallest are typically deletions of this locus control region, but they can also, we will see that in further slides, extend into the genes. Deletions focusing here on the locus control region are actually the most common cause in patients originating from the US. When we do our tests and test for the locus control region, often we're like almost done, diagnosis is clear. These deletions come in different sizes. As said, the minimal situation typically is a deletion of the locus control region, but we also have cases where the deletion extends within the red opsin gene or also further into the green opsin genes, or we have also seen patients that have an intact locus control region but deletion of all genes, and also intragenic mutations have been identified. The second mutations I want to introduce are pathogenic point mutations, so missense mutations, frame shifting mutations, nonsense mutations, and typically they are found either if there's a single gene left, hybrid gene, or a single red gene within the cluster that in addition carries such a point mutation, or if there is a structurally intact opsin gene cluster, then every single copy of the gene typically carries the very same mutation. This is surprising maybe, but it is fact. And we believe that this is due to gene conversion, so that mutations move through the genes, so to say. The most common mutation by far that is observed is a missense amino acid substitution, cysteine 203 arginine. This is by far the most common cause of BCM in European patients. Other missense mutations have been observed, but they are really rare, and I want to state that many of them are really private to single families. The third important mutation type are the so-called exon 3 interchange haplotypes, and again, they result in BCM if they affect a single red or red-green hybrid gene or are present in every gene in a larger cluster. They have puzzled us for quite some time. It was a long story. Exon 3 had long been known to carry certain common polymorphisms that are spread in the normal population, but it was observed that what we thought at that time, that certain amino acid combinations seemed to be linked to BCM or Bornholm eye disease, and they had been acknowledged as LIAVA and LVAVA, so on the protein level sequence. But actually they are, of course, due to polymorphisms on nucleotide level. And it was only in 2012 that Yzer and Uyama and co-workers suggested and proved that these exon 3 interchange haplotypes actually result in exon 3 skipping, resulting in complete loss of functional opsin if such deleterious exon 3 interchange haplotypes are present. And by now, about 130 of such exon 3 interchange haplotypes have been functionally characterized. Some of them result in complete missplicing of the affected opsin. Some result in partial missplicing, and some haplotypes present with normal wild-type like splicing. Currently, we consider interchange haplotypes resulting in less than 25% of correctly spliced opsin transcript as deleterious, but there is no functional proof or evidence to that. This is a line that more or less we have set for ourselves. Now, you may wonder why BCM testing is rarely available in diagnostic genetic labs. In former times, when testing was based on an exon by exon PCR and sequencing approaches, this was hampered by this quite complex structure of the opsin gene cluster and this high sequence conservation. And this makes it a bit difficult for labs to establish such a test and also get it through the certification. Then, of course, the exome and genome-based short-read sequencing technologies evolved, and people, of course, had high hopes that mapping would allow that BCM is tested in diagnostic labs. But again, this high sequence identity results in the situation that the short reads are not mapped correctly to the OPN1LW or MW gene. And in addition, many labs work with exome-based pipelines, and the locus control region, which is important for, or which is deleted in a large portion of patients, is not enriched by the exome-based approaches. It is possible to read out those NGS data, but it often depends on that you manually inspect more or less your exome or genome-based sequences. And if you have this classical situation that the very same mutation is causative in all genes of the cluster or in such a single gene, they can be read out, and they are sometimes then also reported by diagnostic labs. But coming back to our setup in Tübingen, Bernd Wissinger was very fascinated by the opsin gene cluster very early on in his research career. And he established already in the 1990 a PCR and RFLP and Sanger sequencing-based approach, with which we have since then tested the patients that are entrusted to our lab. We first check for the structural integrity of the opsin cluster. We have specific PCRs in comparison to another fragment. So this is a duplex PCR approach where we test for the presence of the locus control region. Then we have a specific PCR where we test for the presence and integrity of the red promoter and the green promoter. And then we test for the presence of a single nucleotide polymorphism in exon 5 that allows us to draw a conclusion on whether, in combination of these PCRs and RFLPs, whether there is a single gene in the cluster, whether this is a single red or a hybrid gene, or whether there are multiple genes in the cluster. Then we continue on and query for this very common missense mutation, also by PCR and RFLP. And if we have indication that this mutation is present, we confirm it by Sanger sequencing. With this approach, typically we can solve 60 to 70% of cases already. If this does not result in a genetic molecular diagnosis, we continue on with our so-called extended screen that is gene position specific. For this, in the very first instance, we amplify the very first gene in the cluster, which is typically or needs to be actually a red opsin gene, and all downstream genes. So we cannot separate the different copies of green, whether there is 1 or 5 or 10. This fragment will always simultaneously amplify all green genes in the cluster. Then we take a very small amount of the
I don't hear Suzanne anymore. Does other people have the problem?
6. Somebody has her micro on. Please turn it off. Simultaneous amplification of exon 6. And then we more or less evaluate the Sanger sequences, search for novel missense, nonsense or frameshift mutations, and draw our conclusion. And as said, novel variants are really very, very rare. Last but not least, we look at exon 3. On the one side, for the red opsin gene, which we call the proximal gene, and on the other side of all green gene copies, so the distal ones. And we read out first the nucleotide level because it's easier to perceive. We translate it into the protein situation. Also, we know the defect is not on the protein level. And then from the functional data that has been gathered within the research community, we then conclude that this very haplotype goes along with, in this case, for example, the LIVVA haplotype. This very one results in 3 to 5% of correctly spliced transcripts, and this is considered deleterious. Or in this case, for example, we have 2 different haplotypes, one in the proximal gene, which is an LVA-VA, which is one of the common function loss haplotypes. And in the green opsin copies, we have an MVA-VA haplotype, which has been shown to roughly result in 20% of correctly spliced opsins. And taking all this in consideration, we then have to decide, is there a genotype that is suggestive of BCM? Or also BED is, of course, so Bornholm eye disease can be a diagnosis. What are the genetic constellations in BCM in comparison to Bornholm eye disease? If all genes within the cluster have a deleterious mutation or haplotype, this is suggestive of a clinical diagnosis of BCM. if we have such a constellation, so there are multiple genes in the cluster, the structure of the cluster is intact, but we have a deleterious mutation or haplotype in the first gene of the cluster, this is typically indicative of a Bornholm-eye disease. And these are the conclusions that we then draw. In this case, a large deletion was identified that would be of the locus control region, which would then result in a BCM phenotype. Here in this case, obviously, an exon 3 haplotype was identified in an intact cluster only in the first gene, and this would be suggestive as of Bornholm-eye disease. But sometimes also we have to state that genetic testing does not support a clinical diagnosis of BCM, and these are, of course, the least nice cases. We sometimes also have to state that the genetic testing results that we have achieved neither support nor exclude that this is a BCM or a BED-like phenotype. And this is the case sometimes for novel missense mutations, unclear yet untested exon 3 interchange haplotypes in the situation if there are multiple genes or genotypes, because sometimes you also have such a situation, you have a multi-gene, the first gene in the cluster is clear, it has the other deleterious genotype, but there are like heterozygous situations in the downstream genes. We do not know at which position the deleterious haplotype is positioned, and then it becomes really difficult. Looking at numbers again to almost finish off, within our cohort of around about 470 BCM patients that we have ever tested, about 40% carry the missense variant cysteine 203 arginine. The second most common cause are the large deletions with about 30%. Exon 3 haplotypes account for about 25%, and other rare frameshift, nonsense, or missense variants only play a minor role. And with this, I will summarize. So keeping in mind, with relation to glucose monochromacy and Bornholm-eye disease, we have to understand that the OPN1, LW, and MW genes are located on the X chromosome as a tandem array. Exonic and nucleotide, intronic nucleotide sequence conservation is super high, almost identical, resulting in non-allelic homologous recombination and gene copy number variability. A normal color-sided male would be fine with one single red, one single green gene, as only the first two genes in the cluster are expected to express, and one cone photoreceptor only expresses one cone pigment. Looking at the situation in blue cone monochromatism, it is a rare X-linked disease caused by structural rearrangements and pathogenic variants in the opsin gene clusters. The most common and important mutation types are these large deletions, the cysteine 203 arginine missense variant, but also other single nucleotide variants, and the exon 3 interchange haplotypes that result in exon 3 skipping. But in that case, if they are related to blue cone monochromatism, this means either there is only a single gene in the cluster carrying this mutation, or all genes in the cluster, or at least the first two genes, need to carry a deleterious phenotype. Otherwise, Bornholm-eye disease needs to be considered. And with this, I'm ending my talk. I want to acknowledge that I took some slides out of Bernd Wissinger's slide deck, and I'm happy to take any questions.
Thank you very much, Suzanne, for this very clear presentation explaining this complex opsin cluster. Are there any questions? So if not, then we can go to the last session, where novel diagnostic approaches will be covered, and this will be presented by myself. My name is Lonneke Haer-Wigman, and I work as a clinical laboratory geneticist at the Department of Human Genetics at Radboudumc in the Netherlands. The Department of Human Genetics at Radboudumc is a leading center for clinical genetics, genetic diagnostics, and research on hereditary diseases. and my diagnostics work focuses on inherited retinal dystrophies, which includes some challenging genetic regions, and my favorite being the Opsin gene cluster, which I will present today. So I will share my presentation. Let's have a check that I'm sharing my presentation. Yes.
Yes, you are.
Yes. So my presentation is divided into 3 parts: current diagnostic genetic testing, Opsin gene cluster diagnostic testing, and future diagnostic genetic testing. First of all, I will discuss the current diagnostic testing, and in many European countries, short-read exome or genome sequencing is performed as a first-year genetic test in patients with inherited retinal dystrophies, as already said before. So because I work in a diagnostic laboratory, I will use 2 clinical cases as a guideline throughout my presentation. And this first case is a 54-year-old male with cone dystrophy and myopia. There was no family history of visual impairment, and we performed short-read genome sequencing with analysis of a gene panel for visual impairment. So in the table of comments, you may already spotted the word short-read and long-read sequencing. Almost all diagnostic laboratories use short-read sequencing. With short-read sequencing, small fragments, typically ranging from 100 to 300 base pairs, are sequenced. And these fragments are then aligned to a reference genome to reconstruct the patient's exome or genome. However, due to the small size of these short-read sequencing has some limitations. For example, genomic regions that contain repetitive sequences, such as the Opsin gene cluster, may be poorly represented or under sequence, as already was mentioned by Susanne Kohl. If we return to our clinical case where short-read genome sequencing was performed, where short-read genome sequencing was performed, we are now looking at the print screen of the software developed by our bioinformatics team for variant analysis. With standard variant analysis, no variants in the Opsin gene cluster will be detected, as already been presented before. So why is this the case? When we look at the sequencing data in IGV, you can see here the Opsin genes at the bottom. In this top bar, you can see the coverage, which represents how many times each base has been sequenced of this genomic region we see below. The higher the vertical line, the more times that position has been sequenced.
I cannot hear you, Lonneke.
I think we have lost her. Does anybody have her telephone number? It might. Lonneke, do you hear us?
I'm 3 days checking.
I'm afraid I don't have her phone number. Sorry.
Susanne Yzer, do you?
Yeah, no, I don't have it, but I'm Franka Hartgers, who is also in this call. She has her number, so hopefully Franka can contact Lonneke.
Perhaps anyone have any questions in the meanwhile
I have now
when we
two.
are waiting?
Yes, I have a question for Susanne Kohl, but this will be probably answered by Lonneke too. My question, Susanne, is if you think that in the future you would rather go for a single-step testing rather than multi-step testing of the Opsin cluster?
So for sure, if once that the long read sequencing reliably work, this for sure will solve the problem. But we have, so Bernd, is Lonneke back? No.
Oh, I'm sorry. No, so one of my colleagues just walked in and she's also thrown out of this meeting. So probably there's something wrong with the link, or does it expired, or I don't know.
It's probably something with hospital network or the network where you are sitting.
Yeah.
Link should work because it's working for everyone. It's rather a problem with network.
Most of the time it works best for me if I go over the, what's the browser of Microsoft called? Maybe you can recommend that to her.
We were trying to get in touch with her.
Somebody.
So I'm sorry, I'll come back to you as soon as I hear from her.
So we can elaborate a little bit on the long distance, long read sequencing. So we are doing some pilots on that, but you really need to have high quality, high molecular weight DNA. And we have been able to enrich fragments. So Bernd does a CRISPR enrichment of about 150 kb in size, and they try to enrich for these. There are some few reads that work, but at this time point, we don't have reliable long read sequencing for the BCM cluster. So we also know that there are some bioinformaticians working more or less on a more reliable read mapping, and this is something that we hope very much that this may be picked up by diagnostic labs, because there's for sure an unmet need for genetic testing in BCM. Very, very few labs report back to the patients, and I also have to admit, also the ones that do a readout from their short read sequencing. Sometimes if patients then come to our clinic and we do our standard screening, we do not come with the same result, because often not all reads are mapped, and then more or less it is reported as if there is like a, we call it uniform or homomorph situation within the cluster, but reality is often then when we do this detailed characterization of the cluster, we end up with a heteromorph situation, meaning that there are different genotypes in the cluster, and our conclusion is often not the identical to the one that is forwarded to us.
Thank you so much, Susanne.
I'm so sorry, but I have no internet anymore on my main computer. So I just got phone calls, so I'm out.
Dr. Lonneke, maybe I can share your presentation on my screen and you can comment if you want.
Yeah, you don't see my, I can share again. I'm now on a different computer. I can try to share again. Do you see my presentation?
No, we don't.
Not yet.
No.
It's really annoying. Oh, yes.
Yeah, Lonneke, I just learned that everyone here is out of internet at our department, except for me apparently, but
Okay, we have no internet at the hospital anymore.
no.
I'll share again. I will share the screen. Webinar. Do you see now? Do you see something of my presentation?
No.
No, okay, then you can share it. Apologies. Oh, this is really frustrating. Because I also don't know how much you already missed.
Only
But
when
we
you
can
started
guide
to
you.
explain more or less the read mapping, then you froze already.
Lucy, can you share, please, Lonneke's presentation?
Okay, I'm on it.
Okay, thank you. Well, the network's problems are out of our control, so we can't affect it.
Yeah, I'm trying that I can make a hotspot from my phone to my computer. Maybe that works. But my computer still says it has internet.
Okay, I think Lucy is just sharing.
Yeah, I can see myself. Okay. Oh, and I want to look at my phone, so it's really small for me to all manage. Can you set it in presenter mode?
Yes, of course.
Yeah, so yeah, I think this is okay to go further on. Now you can next slide, please. Next slide. I will just explain this.
We didn't have that. We didn't have that.
Okay, then go back. Just then we'll have some delay in the program. So here, so this is the visualization of the data within the normal data, and below you can see the opsin genes. On top you can see the coverage, and the length of the vertical lines depict how many times this position has been sequenced. So this is the coverage. You can see some small dips in the coverage, but it's sufficient for variant calling. So the reason why no opsin variants are called in the normal standard analysis is not due to coverage problems. So now you can go to the next slide. So if you now zoom into the OPN1-LW gene, you can see these small bars overall. And in the beginning of the gene, before the OPN1-LW gene, these bars are gray that indicate high quality reads. But if you then look over to the OPN1-LW gene, you see all these bars are white. This is because they're labeled as low quality. Next slide, please. So now we zoom in even further on exon 3, and then we can see 4 variants in this exon 3. You can see that because of the colored vertical lines and also because of the letters within the reads. And these letters are all in white reads, so it means that these reads have low quality, and variants in low quality reads will not be called. So next, not slide, but next, yeah. And these reads get a low quality because of the high homology or similarity between the OPN1-LW and OPN1-MW gene, as already was explained by Suzanne. Next slide, please. The variants are present in the data, but they have not been called. And to address this, our bioinformatics team developed a specialized tool called the SEC-DUP tool for analyzing variants in highly homologous regions in short-read sequencing data. So next slide. So now we're looking at the results of the SEC-DUP tool in our variant analysis software. And in the analysis of case 1, 2 pathogenic variants or class 5 variants, as you can see by the 2 arrows, are detected in the OPN1-LW gene by our SEC-DUP tool. Next slide. So how does this SEC-DUP tool actually work? This tool uses an artificial reference genome, which is based on the standard HT38 reference genome. However, nearly the entire genome is removed and only one segment from a segmental duplicated region is retained. For example, in the Opsin gene cluster, all OPN1 genes were removed. No, sorry, I had again problems. But only the OPN1-LW gene is left over. You press enter, please. In this way, all reads from the Opsin gene cluster are forced to align to the OPN1-LW gene. Next slide, please. So if you now zoom in again on exon 3, we can see the same variants as before. But with our artificial SEC-DUP reference genomes, the reads are gray, marked as high quality, and now the variants are called, as we already saw before. Enter, please. However, we still need to perform a second independent test to know the true location of these variants, because in this analysis, all variants were forced to align to the OPN1-LW gene, also the reads that originated from the OPN1-MW gene. Next slide, please. So how often are causal variants detected in the Opsin gene cluster when only a broad genetic test as exome or genome sequencing is requested? In the 15 months, we have performed 573 genomes with the analysis of the gene panel for visual impairment. These analyses are performed on patients with retinal dystrophy, but also in patients with optic atrophy, cataract, myopia, etc. So it's really a broad patient panel. However, we still find in 10 patients causal variants in the OPN1L in the Opsin gene cluster, and of which 3 were female. In all patients, the causal variants that we detected were combinations of variants in exon 3 that disrupt splicing. Next slide, please. So in the second part of my presentation, I will discuss the specific test for the Opsin gene cluster that our laboratory has developed and which has been published. Next slide, please. So we're still with clinical case 1, and we are in need of a second independent diagnostic test. The test we have developed includes an MLPA assay to determine gene copy numbers and long-read amplicon sequencing for detecting variants. Next slide. So why do we need this copy number analysis? Now, Susanne Kohl already explains this really nicely. The copy number of OPN1-MW is highly variable among individuals. And long-read amplicon sequencing provides a good estimate of how many OPN1-MW genes are present, but estimating is, of course, not a very diagnostic approach. Copy number analysis is generally possible with standard exome and genome sequencing. However, this does not work for the Opsin gene cluster, especially when HC38 is used as the reference genome. Next slide, please. The MLPA for the Opsin locus was developed together with MRC Holland, and you can find more information on their website. I won't go into any technical details of how MLPA works, but here is the result of our clinical case. And thanks to my great technicians, I always get a really nice overview of the results as shown in the middle at the bottom. Here you can see at one glance that 1 OPN1-LW gene is present and 2 OPN1-MW genes are present, of which 1 is a hybrid gene. Next slide, please. After determining copy numbers, we perform long-read amplicon sequencing. An amplicon is simply a piece of DNA that has been copied during PCR. After amplification, the amplicon is sequenced with PacBio Hi-Fi long-read sequencing. Using PacBio Hi-Fi, we can sequence fragments up to 60,000 base pairs. And if you forgot, short-read sequencing mostly goes up to a few hundred base pairs. For the Opsin cluster, we developed multiple amplicons. Next slide, please. Next slide. 1 amplicon for the first gene and 1 for the second and any consecutive Opsin genes, as also already Suzanne explained. So if 2 Opsin genes are present, as shown in B, these 2 amplicons can determine the complete composition of the locus. But in case with 3 or more Opsin genes, with 3 Opsin genes as shown in C, these 2 amplicons will not be able to determine the complete composition of the cluster, because it's not known which of the OPN1-MW genes is in the middle and which is in the last, because these gene copies will be amplified and sequenced in the same reaction. Next, please. As only the first 2 gene copies of the Opsin cluster are clinically relevant, as Suzanne also mentioned, it is of importance to determine the complete composition. And to address this, we have developed a third amplicon that's specific to the last gene in the cluster in cases with 3 Opsin genes. Next slide, please. These are the sequencing results of clinical case 1. And with MLPA, we determined that 3 Opsin genes were present, and therefore all 3 amplicons were amplified and sequenced. And also, without going into details, the top bar shows an OPN1-LW gene with a LAVAVA combination. The second bar shows an OPN1-MW gene with a LAVAVA combination, but also 1 hybrid OPN1-MW gene. And the third bottom bar shows the amplicon for the last gene, and it shows that the last gene in the cluster is a hybrid OPN1-MW gene. Next slide, please. And when we take these results together, the pathogenic LAVAVA combination of variants is present in both the first in the OPN1-LW gene and the first OPN1-MW gene, and the last gene of the clinically irrelevant gene in the cluster is a hybrid OPN1-MW gene. And as said before, this LAVAVA combination strongly disrupts the splicing of exon 3 and is therefore pathogenic. Next slide, please. So these variants are the cause of the myopia and cone dystrophy of clinical case 1, and most likely this is not a cone dystrophy but a blue cone monochromacy patient. Next slide, please. To conclude on the specific test for the Opsin gene cluster, between January 2015 and June 2023, our laboratory conducted the Opsin-specific diagnostic test in a total of 155 male patients. Of these, 96 had their clinical diagnosis genetically confirmed. On the other hand, for 47 patients, the clinical diagnosis was not genetically confirmed. In 12 cases, it was unclear whether the clinical diagnosis had been confirmed genetically. This uncertainty was due either to the presence of variants of uncertain significance, or VUS for short, in 11 patients, or because we were unable to determine the complete composition of the Opsin gene cluster, which was the case in 1 patient. Next slide, please. Up to now, I've covered current diagnostic testing, but let's look at the future of diagnostic, long-read genome sequencing. I'm very fortunate that for me, this future is already here. Starting from October this year, our diagnostic laboratory performs long-read genome sequencing as a first-tier genetic test for patients with visual impairment. I should note, however, that this diagnostic test is currently only available when requested within the Netherlands and cannot be requested from abroad. Next slide, please. We start with the second clinical case, a 30-year-old female with myopia. Her father had high myopia and reduced vision. Long-read whole genome sequencing with analysis of the gene panel for visual impairment was performed. Next slide, please. In this case, standard variant analysis does already show variants in the OPN1-LW gene, as can seen by the 2 arrows. Next slide, please. Unfortunately, when we look at the data in IGV, this data doesn't make any sense. This is due to the HD38 reference genome. Next, please. For the Opsin locus, it is as if somebody took scissors and glue, cutting pieces from different genomes and pasting them together to form this locus in the HD38 reference genome. As a result, the HD30 reference genome does not accurately reflect the reality of individual genomes. Next, please. Next, please, sorry. Yes. A de novo assembly, an unbiased assembly without the use of a reference genome, would be better for the Opsin gene cluster. Next. We performed a de novo assembly in 2 other clinical patients. Both are males with 1 OPN1-LW gene and 2 OPN1-MW genes. As they don't have a third OPN1-MW gene, you see a gap in the sequence if you compare their sequence compared to the HD38 reference sequence. Next, please. Unfortunately, a de novo assembly is very time-consuming and not feasible for every diagnostic patient at this moment. Next, please. Luckily, there is a specialized tool called Paraphase for hi-fi long-read sequencing data. This tool can accurately identify and phase variants in challenging, highly similar gene clusters. Next, please. Paraphase performs a similar trick as our SecDub tool by mapping all reads to the OPN1-LW gene. It then constructs the different haplotypes representing the Opsin gene copies and finally determines the order of these haplotypes within the cluster. And next, please. If you zoom in again on exon 3 of our clinical case 2, variants are visible and they are in great quality reads. Thus, these variants will be called, as we already saw. On the left, you can see that 5 different haplotypes have been detected, which means that 5 Opsin genes are present in this clinical case. Next, please. Paraphase also includes a report that provides detailed information about the Opsin gene cluster. Since all the information comes as one long string of text in our variant analysis software, I've created an overview of some of the information that's present in the report. Next, please. First, the total, the OPN1-LW and the OPN1-MW copy numbers are given. Our clinical case has 2 OPN1-LW genes and 3 OPN1-M gene copies, which is consistent with what is often found in females. Next. Second, paraphase determines which haplotypes/genes are present as first genes, which are the last, and the genes that are left over will be the middle copy genes. Paraphase also determines whether these genes are OPN1-LW or OPN1M gene copies. In our patients, the first genes are 2 OPN1-LW gene copies, while the last and middle ones are all called as OPN-MW gene copies, what you also expect. Next, please. Third, paraphase also determines the variant combinations of exon 3. In our clinical cases, the pathogenic LAVAVA combination of variants was detected in one of the OPN1-LW genes. Next, please. Our patient has thus an heterozygous pathogenic LAVAVA combination in one of the OPN1-LW genes and no pathogenic variants in the other genes. Next, please. Our clinical case is therefore a carrier of born-home eye disease. Carriers of born-home eye disease may also exhibit symptoms such as high myopia, and these symptoms are often milder than observed in affected males. Next, please. We analyzed the paraphase data of 78 opsin gene clusters from control samples. Opsin gene clusters with one OPN1-LW gene or with 1 or 2 OPN1-MW genes were most prevalent. Moreover, we looked at the combination of variants in exon 3 and didn't detect any pathogenic combination of variants in this first subset of data. Next, please. To wrap up my presentation, in short, with exome sequencing and genome sequencing, detection of pathogenic variants in the opsin gene cluster is possible. However, a specific tool designed for homologous regions is necessary for automated variant detection, and confirmation with a second independent genetic test is required. In long-read genome sequencing, detection of pathogenic variants in the opsin gene cluster is possible. However, still a specific tool for this region is necessary. However, this tool is already supplied by PacBio itself. And often independent confirmation is not needed with long-read sequencing due to the high resolution of long reads. Next, please. My last slide contains some general information. If you are in need for a diagnostic test for a patient with a differential diagnosis of born-home eye disease, glaucoma, monochromacy, or X-linked cone dystrophy with myopia, and your country does not order this test or does not order this diagnostically, you can order this specific test I presented today on our website. And if you want to have more information on, for instance, our bioinformatic tools or other laboratory topics, every summer our laboratory hosts a summer school, which is free of charge. And for more information, you can email me. Yes, next slide. Thank you for your attention. And are there any questions? Sorry for the delay and the problems with the internet. So I can also not see the chat anymore. I'm really helpless here. I'm so sorry.
No problem. I will have a look at it.
I think there were no questions in the chat. Yeah.
I have a question, if I may. And thank you very much. Very nice presentation. I have a question about the long read from PacBio. So the reads are shorter than one gene repeat. So are you confident that you have enough variations in the different copies to fully reconstruct the whole genotype for every person?
So it's not always possible, and in females it's more difficult than in males. So also PacBio itself sometimes says, okay, I'm not possible to find the next gene. It really depends on the overlap, because indeed the reads are mostly as long as the gene itself, and if it starts halfway, often it can go to the next gene and then it can face each other. So it's possible, but not in all cases. But the software itself says when it is able to face the complete locus, or that or only part of the locus. So the first copy is always quite easily done, and also the last copy, but sometimes it has difficult to determine the middle copy.
Thank you.
I have a question on behalf of patients, and I would like to ask what will be the future, in your opinion, Lonneke and Suzanne, what will be the future of DNA tests for glaucoma monochromacy in 10, 20 years?
I think it doesn't take that long. I'm very convinced that the long-read sequencing, so also Tübingen here, our diagnostic unit, have set it up. And I've seen the first reports now too, and Bernd is looking into these data. Those countries and labs that can offer long-read sequencing, Renata, I'm quite convinced that they will also be able to report this within the next few years, I hope. Maybe even earlier, we'll have to see. What's your opinion, Lonneke?
No, I agree. I think long-read sequencing is the future for all diagnostic labs. I only wonder whether it's really an expensive test. And with exome sequencing, you can already find, I skipped that part because my presentation was too long, 85% of variants in retinal diseases. And as you saw in my presentation, there is some misdiagnosis sometimes. So then long-read sequencing will be, I think it's the future. And I hope the cost will go down so that we can provide it in every country. But at this moment, also exome and genome sequencing is not provided in every country. So especially for such a specific disease, and I think which is clinically, there are some differences compared to other retinal dystrophies, should be recognizable. And then you can go for specific tests or really request long-read genome sequencing as your first tier test instead of other tests. But I agree, it will be long-read sequencing. And this is just the first results and the first versions of the data analysis software. And we saw that also with exome and genome sequencing, they will improve. So it will get better within the times. And I hope really when we use a different reference genome, the telomere to telomere genome, it makes far more sense. But AC38 is really horrible for the Opsin gene cluster.
Thank you.
Lonneke, we have a few questions in the chat. The first question is if it's possible to have tests for persons in Sweden within your lab.
Yeah, I work at the diagnostic lab, so you can request all our tests online for our website. So that's possible. You can always email me what specific. So the specific test is not a problem. For the broad visual impairment test for abroad, we at this moment still use exome sequencing, but we will change that to genome sequencing, I hope within a few months. And we always use the SecDub pipeline for all our cases, but we do for people abroad, we do our testing. So that's not a problem. You can request it.
I just want to say, so I think this question was asked by probably the mother of two children who are affected. So I believe they need to go to the ophthalmology, who is then contacting genetic laboratory, and genetic laboratory is contacting Lonneke and requesting this test. So that would be the pathway to go.
Indeed. Sorry.
So in our research setting, you know, and Renata has mentioned that they support us for research testing, and we offer that also in direct patient contact. They only have to supply us with copies of reports, and of course the consent forms that we forward on.
So we, as a diagnostic lab, we really need the request from a clinician. It can be a clinical geneticist or an ophthalmologist. Yeah, that's the difference.
And the second question is PacBio the best approach, or do you think that nanopore is still on the table given the longer reads?
It's also part quality. So PacBio now has higher quality than Export Nanopore, but Export Nanopore is always also improving in that part. I think both are you can use both in the future. I think for this moment, PacBio is a bit better because of the higher quality, which is really necessary for Opsin genes because they're so similar. But I think in the end, both will be possible. But yeah, our laboratory really invested into PacBio, and also for the Oxford Nanopore, especially for the assembly. If you use a reference genome, you also need another tool to really look into the haplotypes, but I'm not really familiar with Oxford Nanopore sequencing.
Elfriede de Beer commented on the request with the long-read sequencing for Sweden that she is aware that long-read sequencing should be available also in Sweden. So maybe it makes sense to contact Karolinska.
Yes, I was pointing toward Karolinska, and I noticed that Karel de Geer is online and he's working at Karolinska Institute.
Yes, we are right now collecting the first few samples to test this new technology, and it will be a PacBio long read that we use.
Great, thank you. I'm the mother that asked the question about this BED test is because I have two sons that are diagnosed with that here in Sweden. So therefore I just asked, I'm just curious because I have three other children, also two daughters and one son. And as far as I know, they aren't not affected right now. So therefore I was just curious. Thank you.
And where are you based? Are you based in Stockholm to access the ophthalmology and Karolinska?
Yes, they're at Karolinska, and we have done all the tests there with EGR, blood tests, and so on for a couple of years ago. So all my family have been testing these EGR and blood tests.
Okay.
So thanks.
I
Thanks
am
for your
aware.
support.
You can contact me on email, and we can follow this up.
Yeah, thanks a lot.
So if there are no more questions, then we can finalize this webinar. And again, my apologies, but the internet is still lacking here. Sorry. Thank you so much for your attention.
Thank you
Thank you
very
for
much.
organizing.
Everybody.
And only just to say, this webinar will be recording will be placed on the ERN-EYE homepage. So anytime you would like to come back and revisit, listen to that again, you are welcome to do so. Okay, thank you very much to all speakers.
Thank you.
Thank you. Have a great evening. Bye.
Bye.
Lonneke, je zit ineens in beeld.